Welcome. Today we are going to look at neutralization test. In neutralization test, the principle rely on the fact that antibodies neutralizes the effects of antigen. You can try to demonstrate. If I have an antigen, so that is antigen, call it K. It may have biological effects, like maybe killing an animal or causing symptoms. When it reacts with specific antibody, now we have antibody K, actually specific antibody K to form a complex that is antigen antibody complex. The biological activity of the antigen is cancelled, so it cannot cause disease or cause death of the animal. So we say it neutralizes the biological activities of an antigen. So that's the principle we use to neutralization test. So there are various substances that are neutralized when they react with their specific antibodies. So such antigens are viruses, poison, and even enzymes. So when they react with their specific antibodies, their biological activities are neutralized. So we are going to start by looking at the virus neutralization test. That's it, by looking at the virus neutralization test. In virus neutralization test, the biological effects or activities of the virus are neutralized when the virus is reacted with its antibody. When the virus is reacted with its antibody. So there are a number of such biological activity. One of them is called cytopathic effect. One of them is called cytopathic effect. Or what we term CPE, cytopathic. Cytopathic effect. In cytopathic effect, the virus normally changes the structure of the cell, structure or morphology of the cell. It can make the cell to look round. Can make the cell to look round instead of having any other shape. It can make the cell to become round, very round, or make a number of cells join together to form a larger cell, or it can cause inclusions in the cytoplasm of a cell. It can cause inclusions in the cytoplasm of a cell or the nucleus. So you see things which look like granules in a cell which do not have granules, or inside the nucleus, which normally may be clear with a few inclusions. So that is what we call cytopathic effect. We call cytopathic effect. Another biological effect of the virus is is agglutination of red blood cells by a virus. We learned about agglutination. So a virus can cause agglutination of particular red blood cells, because some virus particles have receptors to red blood cells. So they will cause red blood cells to touch onto them using those receptors, forming a clump. So these are red blood cells. This is a virus, RBC. So when the clump grows bigger, it forms agglutination. So that is called agglutination or hemoagglutination of the red blood cell by a virus. It is not an antigen-antibody reaction, but the virus have receptors for the red blood cells. So it makes them attach on the virus, resulting into clumping, which is called hemoagglutination. So we are going to start by looking at neutralization of cytopathic effect, and the neutralization of the cytopathic effect. Before we do that, I will explain what we mean by cell line, because we won't talk about it. You have grown bacteria in a media. You introduce bacteria in agar containing nutrients, and the bacteria grow into colonies. The bacteria grow into colonies. When you want to grow other cells, which are not bacterial cells, then we normally use a kind of media which is not solid, normally in a conical flask. It's liquid media. So once we have that liquid media, then we introduce cells onto them. The cells grow normally on top. Cells. So when you want to grow eukaryotic cells, like cells of the liver or cells of the kidney, then you use a media which contain nutrients, normally in solution form in a conical flask. Then grow cells on top of the cells grow on the surface. They will be individual cells, but they form a layer on the surface. That's what we call cell line. So these cells can be used to demonstrate cytopathic effect. So you inoculate the virus inside these cells. So you introduce the virus. Just as you reduce, once you have inoculated these cells, then you inoculate the virus and then observe what are the effect of the virus. That can be done, and then you see cytopathic effect. So that's what we call a cell line. So when you talk about it, you will not have a lot of problems understanding what we are talking about. So let's start by looking at neutralization of cytopathic effect. So a number of viruses, like poliovirus, can infect an individual. So when they infect an individual, then the antibody against them is produced. That antibody will neutralize the biological effect of poliovirus. So If you suspect that a person is infected by polio virus, then you'll take the serum from an individual, then put in a tube, so that's the serum. So you know that the serum contains the antibody against polio virus. Put it there. Then come up with a suspension of the polio virus. So that's now the antigen. It can be commercial or can be made in the lab. Then add it to the serum, which contains the antibody. This is the polio, suspension of polio virus. Then add it to the serum. Then incubate. All this reaction will be incubated. After incubation, you'll take out the suspension of the virus. So you take the suspension, of the virus and inoculate it in the cell line. So you inoculate it here in the cell line. Then after that, this thing is normally put on a microscopic stage and viewed by a microscope. Then you view the cells after incubation of the virus suspension with the cell line. First inoculate the virus, the suspension of the virus and the serum into the media containing the cell line. These cells must be susceptible to the polio virus so that you can see cytopathic effect. After inoculation, take it out and observe under microscope. If you see cytopathic effects, then you did not have the antibody of polio in the person's serum. That is negative. But if there is no cytopathic effect, then the serum and the antibody which reacted with the polio virus neutralizing its biological activity. So that is clear again. No cytopathic effect is a positive test. Presence of cytopathic effect is a negative test. Then we move on to hemoagglutination inhibition. Again, if you suspect that the body of an individual has a particular virus, then you take the serum. And put in a tube. So then add the virus. This is now the antigen. This is the serum. Not now writing antibody antigen because you know the serum contains the antibody against this particular virus. But this virus you are adding is commercial because the virus in the individual you have not picked. You only picked a serum which contains suspected antibody. So you come with the commercial virus, add it, incubate. After incubation, start the suspension, put in another tube. This now is virus plus serum. Then now add susceptible RBC, red blood cells. Then again incubate. Then observe. If you see agglutination, that is a negative result. So that shows that the virus was not neutralized. There was no antibody to the virus in the serum. But if you don't see agglutination, then that's a positive result, because there was antibody for the virus in the serum which neutralized the virus, and therefore hemagglutination was inhibited. So then we are through with the virus neutralization test. Now we move to the toxin neutralization test. So a number of microorganisms produce toxin known as ectotoxin, because this toxin work outside the bacteria cell. Like the bacteria infect you and it produces toxin outside its body which affect you. You know this because you do microbiology. Endotoxin can only affect you if the bacteria is crushed or killed to produce the endotoxin. But exotoxin are actively secreted by the bacteria to affect the host. So these exotoxin are antigenic. So we can isolate the toxin. We can isolate the toxin. I can isolate the toxin, then put it in a tube. So that the toxin then add the antibody specific to the toxin. If they are specific, the effect of the toxin will be neutralized. The effect of the toxin will be neutralized. So then you take the suspension of antibody plus toxin. Well, let's say this is serum. Not if the antibody was serum, I just use the serum, so that's serum. Let's forget it because it may not contain the antibody required. So against the toxin. So then once you take that, then you put in another tube. And let's say you put in another tube. With an indicator of the biological effect of the toxin. So once you add it here, the mixture, if the indicator shows the presence of toxin, then you are sure the serum did not have the antitoxin, the antibody against toxin, so it was neutralized. But if the indicator does not show the presence of the toxin, then the serum contains the antibody which neutralizes the toxin, and therefore the effect is not seen. An easier way of showing that the toxin is present is to use an animal susceptible to the toxin. An animal susceptible to the toxin. So let's now have a case where we have a tube, we extract the serum. You think a suspected antibody, then we add the poison. Then we add the poison. Now we have the antibody and we have the antigen. The other side was the reverse. We had seen earlier that you can do reverse. You can test for the antibody using the antigen, or test for the antigen using the antibody. So once you add the incubate, then you take this suspension of poison plus serum mixture, then inject it into susceptible animal. So you expect two things. This animal will die because of the poison, let's just a mice, or it will remain if the poison, the biological effect of the poison is neutralized. So if the serum has the antibody against, that poison, then after incubation, this thing will be neutralized. The poison will be, the biological effect will be neutralized. So when you add this mixture or inject it into the animal will not die because the poison has been neutralized. But if the serum has no antibody, then the poison effect, biological effect, will remain and will kill the animal. So a positive test shows that the animal is not killed. A negative test, the animal dies. Then finally, we go to enzyme inhibition test. Enzyme effects are normally shown by reacting the enzymes with their substrate. The enzyme is supposed to be the substrate mainly to produce color change. So that's what we can see. Or the substrate is consumed so that we see it disappears. So in enzyme inhibition, the normal reaction of enzyme and its normal substrate is inhibited, so you don't see it. So what happens, like in the first cases, just have a tube, have your serum, add the antigen to the enzyme, incubate, withdraw the mixture, enzyme plus serum. If this serum contains suspected antibody against the enzyme, then the enzyme activity is neutralized. So you add this to a substrate again in a tube. So this is the substrate. So if you expect color change when you add the enzyme to the mixture, then you should see that color change when you add enzyme plus serum. So when you add enzyme plus serum into the mixture and you see effect on the substrate, then that means the serum has no antibody, because the antibody expects to neutralize the enzyme. But if you don't see that color change, then you know the serum has the enzyme. The serum has the antibody against the enzyme. So it will neutralize using incubation. So when you add it, there's no effect. So enzyme diffusion test is done like that. Thank you, and let's meet next time.